Thank you very much for the introduction, and good morning to Barcelona. So I'm going to give a short company presentation today about Ray Therapeutics. We are a Bay Area-based biotechnology company. We're late-stage preclinical, and we hope to have our lead asset in the clinic by the end of this year. Our mission as a company is to restore vision to patients with all forms of retinal degeneration, including macular degeneration, the dry form, so rare disease as well as large market indications. We have several assets. The latest in development is RTX-015. We are an optogenetics or a visual optogenetics company, so we're developing vision-restoring gene therapeutics. And our big differentiation here is that for the first time, although there's been some early successes with optogenetics, for the first time we have a bioengineered optogenetic medicine which has been optimized for human vision. And with that, we're able to establish higher visual functions than we've seen before, which has largely been limited to improvements in light sensitivity in blind patients, to now the realistic prospect of them regaining some form of higher visual function like visual acuity, contrast sensitivity, and motion vision. All the other prior attempts with optogenetics have required light-enhancing eyewear to drive enough light in, whereas our optogenetic approach works in ambient lighting. One of the huge differentiations that we have as well is our management team, more than 100 years of biotechnology experience with successful exits, particularly in regenerative medicine. And the reason we were able to draw together such an experienced management team is that we really feel this program has been hugely de-risked. As I mentioned, the clinical data with first-generation approaches, but also the fact that all aspects of our clinical and preclinical development have been de-risked. So just a few words about how optogenetics or visual optogenetics actually might fit into the treatment algorithm for rare diseases like retinitis pigmentosa and in other more common diseases like geographic atrophy caused by age-related macular degeneration. So the schematic at the top on the left-hand side shows a cross-section through the retina. I wish I had a pointer, but in the normal retina, you have the photoreceptors that receive light at the top there, and then they relay to other neurons, eventually getting the visual signal to the brain. In pretty much all of the retinal degenerations, you lose the photoreceptors and they don't recover. But you are left with retinal ganglion cells at the end, and we hope to make those light sensitive with optogenetic therapy. For most of the therapies that are in development at the moment, like gene replacement approaches and gene editing, we have to realize that you need the cells, like the photoreceptors, to be there for them to actually work. And so this actually leaves a large proportion of the population who don't have photoreceptors open to optogenetic therapy, and there's not really much there that can compete. One of the really interesting things about our approach, I think, is that it's gene independent. So we don't need to know the underlying mutation or the gene that causes it. It should work for essentially all comers, which broadens the market and the commercial applicability. So this is our pipeline, as I mentioned, optogenetic therapy for retinitis pigmentosa first, with a segue into other inherited retinal diseases, and then hopefully into geographic atrophy. As you can see, sizable, treatable, addressable market sizes in the US, quoted there, 5x approximately for global prevalence of those particular diseases. So just a few words about actually optogenetics then. What exactly is it? So it's actually about the 20th anniversary of the discovery of channel rhodopsins. These are microbial transmembrane proteins. They're channel proteins, and they're unique in nature in that they actually have the ability to be gated by light. So light activates the channel, and you get an ion flux. And these are proven to be seminal in mapping brain circuits. From a visual optogenetics perspective, you have to realize that the retina itself is actually a neuronal structure. It's an extension of the brain. It's directly connected. And in this instance, we are, those cells, as I mentioned, that remain, we are reanimating them as if they were like photoreceptors with optogenetics to restore vision. And this whole technology has been largely de-risked by multiple independent laboratories and, I said, early stage clinical development. So it is about the 20th anniversary. You can see the timeline here of development of optogenetic therapy for vision restoration starting in 2003. We hope to be in clinic by the end of 2023. We've been certainly spurred by the pioneering work of others. There was a publication a year or so back in Nature Medicine of the partial recovery of a patient who was profoundly blind following optogenetic therapy while wearing those light-intensifying goggles, but nonetheless a really amazing proof of concept, and subsequent patients have also been reported for this technology to work. So it could potentially be very disruptive for the field. So a few details about our actual product, RTX-015, which will be the first-to-market, first-in-class precision optogenetic medicine. Our optogenetic protein we call CROWN. So the human opsins in your retinas that you're actually looking at these screens with, CROWN is about as sensitive throughout that range of vision that you're currently using right now. It has a broad wavelength of activation in the visible spectrum with a very large dynamic range from dim lighting like this right up to bright sunlight, and it retains fast switching for human vision. So the realistic prospect of visual acuity, contrast sensitivity, navigation, vision. Yes, it's intravitreally, so it's a simple eye procedure to deliver this using an advanced next-generation AAV-based gene therapeutic targeting those retinal ganglion cells which survive into the disease. Because we're looking for a visual improvement and early, really do think that the approval pathway particularly in RP, a rare disease, should be very efficient. And we have a large commercial opportunity, as I mentioned, with many thousands of patients and a nice comparable with the approved ocular gene therapy, Luxturna, as our comparator. So what of our actual data? So all of the data I'm going to show you is actually generated in vivo in very much a gold standard mouse model system for looking at behavioral responses and vision. As shown on the left-hand side, using an exquisitely sensitive, translatable mouse model with a lot of evidence to support the fact that the findings here should translate into humans. As you can see on the right-hand side in the figure, the early generation optogenetics shown in black were very insensitive to light, whereas CROWN is very, very sensitive to light. 1e13 photons per centimeter per second. It is about dim street lighting at nighttime. So a very significant improvement over other optogenetic approaches. The ones shown here are the ones we actually recreated that have been used in clinic already in early phase clinical trials. And CROWN is more than 100 to 1,000-fold more light sensitive, and thus would be the only one that will not require the light intensifying goggles. This is the data that actually formed the company, really. Light sensitivity is just the most basic of visual functions. Visual acuity as humans and contrast sensitivity are major parameters, of course, for our visual function. When we look at the normal mouse shown in black, you can see that they achieve a reasonable visual acuity. In this particular mouse model, they have absolutely no visual responses at all. They're completely blind. However, when they're treated with the CROWN optogenetic protein, they get more than 50% restoration or even creation of vision in these blind mice. So we've never seen that before with any of the other optogenetic approaches. And you get near normal levels of contrast sensitivity as well. So that's the preclinical data. In terms of everything else that's de-risked, we really have gone, I think, to a level here which gives us a lot of confidence moving into the clinic of near-term success. We're very happy to have partnered with Forge Biologics, who are here. They're doing a tremendous job in terms of de-risking our manufacturing with a commercial manufacturing process, going straight into suspension with no need for scale-up because of the rare disease nature and the small volumes used for ocular gene therapy development. Our clinical development, as I mentioned, is very de-risked with fast readouts, which is unusual for a rare disease, but a huge advantage. The other major advantage we have is that our treatment population is also the same as our safety population, so we can enroll patients who are likely to respond in early phase clinical development. And as I mentioned earlier, we are likely to see early efficacy within a couple of months potentially. So that's the overview of our lead asset. We do have other assets in earlier stage development, which I think are exciting. But hopefully we're going to realize the full potential of optogenetics for vision restoration and substantially improve the quality of life for patients for whom there are currently no available therapies. Thank you very much for your attention.